Hi everyone, I'd like to welcome everyone to the ARC Training Centre and Predictive Breeding seminar series. It's my great pleasure to have Lee Hickey here today, who's going to present on AI in breeding. But first I'd like to do the acknowledgement of country. The ARC Training Centre in Predictive Breeding acknowledges the traditional owners and their custodianship of the lands in which we meet, and that's the Turrbal and Yurruga peoples. And we pay our respect to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. I'm Melanie Wilkinson. I'm a research fellow in the COE for Plant Success, as well as the Training Centre for Predictive Breeding, and also the crop science in coffee. So today's seminar is going to run from 3 to 4, and after the presentation, we're going to have a Q&A session. So we need to make sure everyone online uses the Q&A tab, so therefore we can upvote good questions within there. So don't use the chat function, basically. So it's just a reminder that this seminar will be recorded. So we have Lee Hickey. He is the ARC Future Fellow, and he's also our Director of the Training Centre for Predictive Breeding. And he's within coffee within the crop science. And can we please have a warm welcome for Lee?
Hey, thanks a lot, Mel, for the introduction. And it's a pleasure to showcase some of our exciting research, exploring applications of AI in plant breeding today from my colleagues and students here at UQ, other researchers here at UQ working in this space, as well as beyond. And I hope today we can also have a little bit of fun as well. This morning, I was actually chatting with ChatGPT, and I was asking ChatGPT, can you show me what a plant breeder looks like in the future who uses AI? And it actually wasn't very exciting at all or surprising. Pretty boring, really. Just a standard plant breeder going about their business, crossing plants in the glasshouse, as you do. And you can see here, this plant breeder, traditional, of course, is very focused, determined, while making the crosses, determined to remove all of the anthers from that flower. Yeah, nothing really surprising. But before I launch into some of the case studies I have today, I thought we should ask you what you think in terms of AI. Is it the future of plant breeding? I really want to know. I want to know your opinion. People online, please use this barcode and you can vote. You've got 5 options. You either strongly agree, you strongly disagree. Are you somewhere in between? Sitting on the fence? What do you think? I know it's a strongly worded question, isn't it? So we'll see what sort of responses they're coming in. They're coming in fast. Keep voting, everyone. Keep voting. Still got time. Ooh, I can see a dynamic going on. There's votes are coming in, looking pretty promising. Has everyone voted? Yeah. Okay, well, let's stop voting. Keep that screen open for later. We'll come back to that. And let's have a look at what the votes are looking like. Well, looking pretty good. So majority have they somewhat agree with that statement, that AI is the future of plant breeding. In what form? Will we coexist with AI? Humans, human plant breeders? So let me give some examples and flavors of some of the research that's going on in this space, and we might revisit this question at the end. Okay. So the first thing I wanted to point out was plant breeding has always been predictive. And in fact, in terms of the evolution of our crops, it's relied on humans making predictions. Whether it be domestication and some of the traits that were selected thousands of years ago, they were predicting that if we kept the seeds of these plants, then it would be easier to harvest them the next season because they didn't shatter and fall on the ground. If we think about land races have evolved over thousands of years, they evolved through a combination of natural selection and artificial selection, where farmers were retaining the most productive seeds and tubers to grow the next year, because they were making a prediction. They were making a prediction that they would grow better. Even when a farmer plants their crop, and this was happening thousands of years ago, they would estimate the right time to sow their crop based on a prediction that would work out well in terms of rainfall and temperature, etc. Those predictions are not always right, but predictions have long been a critical part of crop evolution and breeding. In fact, today, we've got more tools at our disposal, advanced statistics in AI and machine learning that we can use to help improve the odds, improve the accuracy of those predictions, particularly when it comes to key points in the breeding process that present bottlenecks or opportunities to improve efficiency. So in my talk today, I'm going to use this as my table of contents and come back to this slide, drawing on some different examples. And here you can see, it's a figure from one of our opinion articles in Nature Biotech with Ben Hayes and some of our early career researchers. And we point out 4 of the key areas where we think AI is promising for plant breeding. The first one at the top there is setting the breeding goal. And while that sounds straightforward, it's becoming more and more complex to get right. We're getting more and more access to data, both from farmers, but also consumers like you and I. And we've got to think about what are our values in terms of the crops that we grow in the future, as well as our preferences, etc. So this seems to be quite a problem well suited to AI. And in fact, I know that some of the large breeding companies are already using AI to help design product profiles that they're pursuing in their breeding programs. But today, the first area I wanted to highlight, some exciting research, is around phenotyping for target traits. And phenotyping is a real bottleneck in breeding, in many breeding programs. Sensing technology, however, is transforming our ability to phenotype plants, crops in the field at scale, at the scale that's needed for plant breeding. And, you know, I've witnessed in just the short time that I've gone from becoming a student to where we stand today, the evolution of these technologies going from fully fledged planes with bottoms cut out with sensors in them to fully fledged helicopters loaded with sensors, a full large-scale helicopter designed by Scott Chapman, to these mobile, fly anywhere, anytime drones fitted with sensors. And that's in a pretty short period of time. Plant breeders are also utilizing satellite imagery for phenotyping at a massive scale. So a lot of things are happening in this space. For our research program, this is the typical drone that we've used a lot, fitted out with a multispectral sensor. This type of technology is enabling us to phenotype traits more precisely. It's taking away the human error in phenotyping for many, many important agronomic traits. Because humans will score a plant differently, whether they're hungry or not, right? This sort of sensor technology is also opening the door for new traits the human eye can't see. So, for example, here you're looking at an aerial view of a field trial using RGB. If we switch to thermal, look what's revealed. We see a strip down the bottom that's irrigated. And it's actually transpiring more because it has access to more water. And so you have a cooler canopy. So canopy temperature is an example of a trait that the human eye could not score and evaluate, opening new possibilities to adapt our crops to different environmental conditions. So I want to shine a spotlight on some research being done by other groups that is really exciting in this space for phenotyping. One of these spotlights is from the Aggie team here at UQ and working on the NVT trials as part of the Inviter project. They've been deploying cameras or sensors on plots all around the country, measuring and accurately quantifying things like leaf area and phenology. And this opens the door for all sorts of applications around G by E by M studies that were just not possible without that sort of accurate phenology data, but also the ability to fine-tune crop growth modeling, like through AppSim, for example. And so it's almost impossible to go to all of these field trials and capture really accurate, precise measurements to this degree. And so it's made possible with this type of technology. Another spotlight I'd like to highlight is, again, from the Aggie team. And this time it's about segmentation of different plant parts using imagery. Now, if we think about many researchers around the world and breeders are trying to optimize crop development. So where are the resources being allocated? In the stems, the leaves, the spikes. This is really important for adaptation. They've developed some really nice pipelines that automatically segments the images into these different plant organs. And so you no longer need to cut biomass with big teams and you can only sample a small number of plots. This technology can scale for hectares of field plots at a time. So opening the door for selection and getting to the genetics of these really important traits. So you can see machine learning for image processing is really powerful. I want to walk you through a case study where we've tried something different. Yes, drones fitted with sensors can see the canopy, right? But can they see underneath the canopy? Can they see the roots? Can they pick up signatures that we could use using machine learning to predict root systems underground? And that's exactly what we did in this case study, because our hypothesis was that the multispectral imaging across the season could capture different biochemical composition or signatures that could be related to genetic differences in root distribution underground. So that's what we're testing. In this case study, we worked with Intergrain, one of Australia's largest barley breeding companies, working on a breeding panel of about 500 spring barleys, genotyped with a SNP chip. And there you can see the population that we're working with. In this study, we conducted trials across 2 years, 22 and 24. And within each of those locations, you can see we had 3 different types of trials going on. In orange, it's the root coring trial where we physically dug up the roots to measure them. And this is like a training data set. We also have the pink section, which is the biomass trial, where we physically did that hard work of cutting those plots, measuring the physical biomass. And in the blue section is our breeding panel. These are the breeding lines that we want to make the predictions on, the predicted traits. Interestingly, we had quite contrasting conditions across those seasons, as confirmed through the AppSim modeling performed by Professor Corinne Chenu and Beta. You can see there that in 2022 it was cooler and there was a lot more rainfall, and that led to higher water availability around flowering and during the grain filling period compared to 2024. So this set up a really nice scenario to compare the roots in these different environments. Here's an overview of our UAV flights across the 2 different seasons. You can see straight away that 2022, we have a lot more flights, almost weekly from time of sowing all the way through to maturity. Compared to 2024, we only had 5 flights at key growth stages. Interestingly, this didn't really affect our prediction accuracy for these traits. And you can see there at the bottom, the timing of some of our biomass cuts for sampling biomass above ground that was used to predict those traits. So digging up the roots, this is the hard work. And thanks to Anton Wasson at CSIRO, we collaborated with him and used his technique called the core break method. And really, this is the most efficient way of measuring roots in the field physically. We take a core, it comes out, and we break the soil up into 10-centimeter intervals, and we count the number of root tips that we see on each side. This provides beautiful data to model the root distribution. Now you'll see we only did this for 20 genotypes, 6 plots, because it took 2 weeks to do with a team of 6 people. You can't do any more. So how can we use this sort of method for this sort of trait in terms of selection, in breeding, or even get to the genetics with that sort of scale? So here you can see the correlation between the root count measurements and root dry biomass. This relationship has already been confirmed in crops like wheat. In fact, you can use machine learning as well to actually analyze those core break sections, and that can take the human error out of the measurements as well. So we ended up with some beautiful data. we start modeling this to model root distribution down the soil profile from 0 to 170 centimeters. You can see there those different colored ribbons are the different genotypes. And interestingly, we actually extracted the number of root counts in those different sections or zones of the soil based on the different soil horizons at that location. That's because the soil properties in those different soil horizons influence root proliferation and growth. So we sum up the root counts in those zones. We also summed up root count for the whole area under the root distribution curve as a proxy for root system size. So a nice way to visualize the differences among the genotypes across the different years is to plot the data like this. And straight away, we can see some genotypes responded across the 2 seasons in a similar way, but others showed quite distinct responses, responding to drier conditions, producing more roots at depth. So these 2 genotypes, for example, could be an adaptive or plastic mechanism that we could harness through plant breeding. Now, these are just 20 genotypes. Now here comes the fun part. So we scaled up we used that training data set to then predict a large breeding panel. And so we tested 2 machine learning approaches, a partial least squares approach and a random forest approach, and they're pretty similar, to be honest. Here you can see some of the results for both seasons, predicting our root traits, our 5 different root traits that we have. And you can see even down to a depth of 170 centimeters, in the test scenarios, we're getting quite good predictions. Some of those predictions are in the order of R-squared values of 0.5, 0.6. So we thought pretty promising. And here's what the heritability looks like for these predicted traits on our breeding panel, capturing a high degree of genetic variance, really promising, up in the order of 0.6 to 0.9 for some of these traits. So really promising for selection or even maybe getting to the genetics. And that's exactly what we did next. We applied our haplotype mapping using our predicted root traits from this method that involves using machine learning as a bridge from a very small data set to a large-scale breeding population. And so we use the local GBV approach to perform our haplotype mapping. So we run a genomic prediction model, backsolve our SNP effects. We then break our chromosome up into segments based on linkage disequilibrium. And each of those blocks, LD blocks, become our blocks. And with the marker configurations within those blocks are different haplotypes. So we sum those marker effects within a block to give us our haplotype effects. And we can identify which blocks are important by looking at the block variance, so the variance in haplotype effects. So to cut to some results, here you can see the 2 different years, 2022, 2024, presented in Circos plots. And I've initially shown only the root traits, the most important blocks for each trait, the top 1% of blocks across the genome for these traits. And you can see straight away the genetic architectures controlling root distribution are different in the different growing environments. If I layer in now across the 7 chromosomes of barley, our blocks that are important for shoot biomass, you can see them in green. And if I layer in the blocks that are important for both shoot biomass and root distribution traits, they're in purple. And that's not surprising because we know that there's master regulators, developmental genes controlling both below and above ground development in barley. But here's the interesting thing. Many of these blocks appear to be quite independent when it comes to controlling or modulating above and below ground development. And this presents an opportunity to deploy these haplotypes into different configurations for different production scenarios. But without the help of machine learning in this case, to scale our phenotyping to the level required to get to these haplotypes, this just wouldn't be possible. Okay, so I'll go to now back to this figure. And the next area I wanted to highlight is prediction. So the traditional approach for prediction is using BLUP, assumes many mutations of small effect based on the infinitesimal model. There are other approaches like Bayesian approaches, which have different assumptions, and these often bring advantages depending on the genetic architecture. But what about machine learning? Does this also offer an opportunity to improve our predictions? And so for this example, it's in sugarcane. We thought that sugarcane would be a good example to test out machine learning because it has a complex genome. And many of the traits that Seema and Ben in the team had been working on appear to have a lot of non-additive genetic variation. So this could be potentially a good test case for machine learning models. Here's the 3 traits that they focused on, and also teaming up with SRA on a beautiful breeding data set, almost 4,000 clones of sugarcane, genotyped with a SNP chip across multiple regions and years. And the prediction we're looking at is the correlation between the prediction and actual performance. Chen Sung evaluated 2 machine learning models, a convolutional neural network approach and a random forest approach. And if you're interested in how he implemented these, you can check out the paper in the plant genome. And to cut to some results, here they are for our 3 traits and the different models. In purple and green are our machine learning models. And you can see where they sit relative to the traditional models in brown. And I'd say that they're almost competitive with those traditional models. And we could probably talk to this slide for about an hour, discussing the possible reasons, etc. But one thing for sure is that it could be limited by data set size, by population size. But probably the most important point I want to make is machine learning is not always the answer. It's not always better. And throwing more and more data to the problem doesn't necessarily improve the outcome. So this is something that I really want you to take home. There's not one single model that's always going to work in every scenario. It depends. To build on this, I think this is a nice segue into another spotlight I'd like to make from our work led by Shunis Chiro and Mark Cooper here at UQ as part of the Center for Plant Success. And this is really interesting because they're using an ensemble prediction, which involves all these different models, traditional and machine learning, coming together to improve the prediction accuracy through that ensemble approach. And so in this example, they used a maze data set on a NAM population for traits like days to enthesis and tiller number, and getting a better outcome for these traits is really quite interesting. Essentially, what these different models are doing are capturing different components of the genetic architecture. They have different weaknesses in terms of capturing all of those different elements, but collectively they're working together to have a better outcome in terms of prediction accuracy. And so here you can see a nice Circos plot that Shuni has created, where you've got these 10 chromosomes of maize. The blue strips represent the different prediction models that he's run independently. What you're seeing is the dark blue, the stronger marker effects that are calculated or estimated from those individual models. So you can see there's differences there with those different models, and they're actually being used in combination to improve the overall accuracy. So if you're interested in this, I certainly am. You can actually check out the new package that's just being released as well, where you can easily implement an ensemble prediction approach. So like I said earlier, it's not just about throwing more data at the problem. And on this, we've got to think about how we think and model biology. And so, and I think the work that's being led by Dr. Owen Powell, Mel Wilkinson, and Mark Cooper is a great example of this, because they're starting to integrate crop growth models with genomic prediction for that exact purpose. And here you can see, if we go from left to right, we've got the traditional approach where we're using those SNP effects to predict the endpoint trait like yield directly. We go a step further across, we start to model the intermediate traits, the secondary traits that could be important for yield, for example, and they're more stable across environments, so it makes sense. But if you go a step further, they're actually starting to model the environmental components, the management components as well. And I think this is a really exciting space because it could potentially improve prediction accuracy across environments, particularly when it comes to G by E by M scenarios that inhibit our ability to use traditional prediction approaches effectively. And on this topic, as a bit of a spotlight, Mel is extending this even further by starting to integrate the AppSim whole genome prediction approach with Ensemble, which is crazy, I think, and really exciting, working with BASF and Iowa State University on a soybean data set where they're starting to model the environmental factors like temperature and photoperiod. And so, the theory here is that it could lead to a much better outcome, starting to model the biology in this Ensemble prediction approach, which is going to be really interesting to see how this works out. So back to my contents slide, and it's the last area I'd like to highlight today, and it's around crossing, the most important part of the breeding process, in my opinion. And so if we think about the traditional approaches for selecting parents, typically we evaluate selection candidates, we test them for yield, disease, quality, and then rank them based on performance, or GBV if you're using genomic prediction. And then we choose the best ones as parents for the next cycle. And through that truncation selection process, it's amazing. It works beautifully well. It drives genetic gain. You get a rapid allelic fixation of those really desirable alleles, but it comes with a trade-off, right? It comes with a trade-off on genetic diversity and a loss of potentially valuable alleles. And so here you can see, for example, line 4, it has low genetic merit for yield, but hidden in the genome is really useful alleles for things like disease resistance that are discarded unknowingly. And so this is a problem. And so with that in mind, it's been a bit of the motivation behind, this FastStack methodology that we've been developing at UQ with Professor Ben Hayes. And so thinking about breeding more at a haplotype level rather than a line level, we could perhaps start to make smarter decisions to keep those valuable haplotypes in the mix. And so I want to highlight that this toolkit is really flexible. There's a few main technologies or methods that are being connected here by humans. And I want to highlight these. So the first step is identifying and estimating our segment effects. Now, I already talked about that. I explained the local GEBV approach for haplotype mapping. It's essentially the same. So we're quantifying the value of a haplotype in the genome. The second step is once we work out what's our target haplotypes that we want to combine through breeding, then we use our genetic algorithm to search across all the possible selection candidates to identify the best set of parents that maximize or combine those target haplotypes. It depends. It could be 10 haplotypes, 50 haplotypes, or a whole genome scale. It really just depends on the breeding objective here. Now, the genetic algorithm is a search algorithm. It's a form of AI, really well suited to this big numbers problem, and picks out the best parents. Then we use our digital twins. So we conduct stochastic genetic simulations to then simulate progeny performance from all those possible cross combinations. So effectively what we're trying to do is identify the most optimal path to bring those haplotypes together in the shortest time frame. And then once we have that path, that crossing path, that's where speed breeding comes in, my favorite technology. And because we just want to make all those crosses as quickly as possible, right? So these tools are really made for each other. So just to visualize what this looks like, this is a nice demonstration. We might have our parents that are being selected by our AI tools. We've quantified the haplotypes across the genome. You can see they're in different combinations, but together these parents have the complete set of desirable haplotypes that we're interested in. In silico, we've designed the ultimate genotype with all those stacked up haplotypes. And so if you're interested in this ultimate genotype, you can read our perspective in Nature Genetics. I'd say it's a call to action for these AI tools, but also the biotech tools that are needed as well to complement AI. So if we think about regions of the genome that need to be recombined, so tools like targeted recombination would be very valuable for this. So just to show how these tools work in action, I got this example on mungbean. We've been working with mungbean breeders across the world through this ACR project, working on this mini core collection, really diverse panel of about 300 mungbean accessions, genotype with the SNP chip. And in this case study, we're focused on seed weight, really important trait for mungbean, for sprouting, etc. And so one of the great things about this project is we've had access to these great data sets through our collaborators around the world. So 14 trials over 4 years. We performed a meta-analysis for this trait. and back-solved our SNP effects. The SNP effects went into our FastStack toolkit, right, as I explained earlier. So let's jump to some results. Here you can see on this axis, we have the GBV for seed weight. And in gray, the gray dots are our mini core collection. So this is our base population, really diverse. And you can see the range in GBVs for seed weight, massive. Now, if we jump over a column, the pink dots are our parents that the AI, the genetic algorithm, has selected. And you can see straight away, those pink dots at the top make sense. They've got high genetic merit, overall genetic merit. Makes sense as their parents. But have a look lower down. We've actually got these parents down the bottom here, which have low genetic merit for seed weight. They're selected as parents. That's because hidden in the genome are haplotypes for seed weight that when recombined with haplotypes in those other parents can actually create these green dots. These green dots are the progeny from those crosses. And specifically, they're double cross F6 progeny. So just 2 rounds of crossing can get us a lift in this trait to that level. But here's the exciting thing. This only takes 18 months. If we do a couple more rounds of crosses, we can actually push those green dots completely off the chart. And that's the exciting stuff. And so we've actually made those crosses through speed breeding, which is fantastic, thanks to Delaney. You can see one of the double cross plants there in action with the really big pods and large seeds. We've performed a seed increase of these FastStack lines, and we're about to send them to mung bean breeders around the world for them to use in their breeding program. So this FastStack toolkit now is being used in a whole range of different crops and traits, which is fantastic because it helps motivate us to continue to develop this methodology and adapt it to different scenarios. And there's plenty of work to do with these tools, which is really exciting. And last, I'd like to spotlight this final piece of work. I visited the team in Beijing and the Chinese Academy of Sciences recently, and this will just blow your mind, because what they've done is they've engineered the morphology of the flowers for tomato and soybean using genome editing, CRISPR, to make them compatible with an AI robot to make the crosses in a speed breeding facility. Right? And here they are. Because a robot can't make the crosses if you have this closed morphology of the flowers, but they've engineered it to be open. They've trained the robot to recognize which are which in terms of flowers. And they've actually shown that the robot crossing is as effective as humans. Crossing is a massive bottleneck in any plant breeding program. So this sort of thinking out of the box and co-designing biology with our technology is really an impressive example, and could help solve some major bottlenecks in the future. So back to this figure. I hope I've convinced you that AI and the application of machine learning, advanced statistics, is really an exciting space, helping us overcome major bottlenecks for plant breeding. And there's a lot of work to be done. I want to jump back to this poll and just see if I've changed your mind now. And we'll see if there's any shift in the stats coming in. Did you vote a B before and now you think it's an A? Or are we getting any movement? I'll give you a preview as the votes come in. There you go. Oh, we're getting a shift. But I would argue that the future is not just about AI, it's also about humans. And it's a coexistence or symbiosis, right, between human plant breeders and AI. And that's really important because we've got to have the right skill sets in our plant breeders for the future to be able to use AI effectively, or at least work in multidisciplinary teams to harness this. So I'm just really pleased. It's made my day. I've seen there's now a strong shift to the left, which is fantastic. I better stop it there. It's dropping the A. So. Fantastic. So we might stop it there. I've got one last surprise for you, a bit of fun. I told you we're going to have a bit of fun today, right? Mm-hmm. And here it is, my special segment. Normally, I get paid a lot of money for this. but I'm going to do it for you guys today for free. And you're very fortunate that I've brought in my crystal ball. You've got to be very careful with this. It's very precious and fragile. It's very powerful as well for predictions. And so what I would like to do is a demonstration of my high prediction accuracy and fortune telling status. I would like to call a volunteer. Let me tell you your fortune, then predict your future. I'm looking for a volunteer, like an early career researcher, maybe doing a PhD or postdoc. Can I see a raise of hands? Everyone's so, oh, yep, you come on down. I'm over here. Okay, now, yep, just, don't touch it. I said it's fragile.
Okay.
You just gotta look into the crystal ball.
Yeah.
And I don't know you, just like a bit closer, like the online viewers. I'm sure they're really interested in this. I don't know you at all, so I'm gonna try to, what's your name? You gotta look into the crystal ball because otherwise it doesn't work. I'm thinking L or Lara.
Laura, Spanish, but you can pronounce Laura.
Oh, okay. Thanks. Oh, that's close. Okay. Let me. I'm just looking into the crystal. You're studying something.
Yeah. I'm a PhD student.
Yeah. Oh, excellent. That's what I thought. Yeah. And on your PhD, you're studying like a fruit or something. It's coming like a berry or some kind of berry.
That's right. Really good. Yeah. I'm working with strawberry.
Strawberry. Yeah. There we go. I told you. It's very powerful. Now, are you worried that AI is going to replace you before you even finish your PhD?
I don't know. Sometimes I think about it, but I have to. I really hope not. Yeah.
Would you like me to predict your future?
Yeah, but yeah, let's go.
Okay, let's have a look. It's the crystal. I can see something. Let's have a look. Oh, now it comes to me. I can see you're in the future. You're a strawberry breeder. You're using AI in your workplace, and you're definitely not being replaced. That's for sure. So I think, and a few dot points are coming to me. I think that definitely there's a coexistence there with AI. And to be successful, some key skills, ability to integrate biological insight with computational power for prediction, really critical. So strong statistics, quantitative genetic skills, ability to collaborate across disciplines is going to be a really important skill for the future for plant breeders, and excellent communication skills, not only with your colleagues and co-workers around multiple disciplines, but also with industry. So there you go. That's the end of my segment. And I want to thank our funding bodies and partners who've been involved in the case studies today. I also want to thank the other researchers who've contributed spotlights that I've shone a light on today. Our training centres are so critical for training the next generation of plant breeders, and GIDC. Thank you very much.
Thank you very much, Lee. That was amazing. You covered so many different topics in breeding and AI, and it was very thought-provoking. So I'm going to hand over to Lee to introduce a wonderful video.
Yeah, so while we have you all here, we're basically launching our very first course as part of the ARC Training Centre. You know, there's been a real demand for courses in this area, both from our industry partners as well as our students and postdocs. And so the first course will be launched in February. It's the first course in a two-part series. The second one will be actually co-coordinated by Mark Cooper and Mel here. The first one is Hannah and I, and we have a short video to explain what the course is all about.
We are pleased to announce that the first course as part of the ARC Training Centre in Predictive Breeding is in the final stages of development. We are launching in February 2026 with our first cohort of the course titled Foundations of Quantitative Breeding.
Our innovative course links theory to practice, teaching breeding principles in real-world breeding program examples. The unique and flexible nature of our course allows you to learn anywhere and at any time, completely at your own pace. But don't worry, we will always be there to support and guide you. Learn from real-world experts in the field with over 20 different perspectives and backgrounds. We are developing a unique combination of content designed to suit every learner. We will use really short theory content, worked examples, and real-world industry breeding case studies designed to engage everybody.
We surveyed more than 60 breeding organisations in Australia, and we found there's a strong demand for industry to upskill their employees in the breeding space. And there's very few options available other than doing a PhD, that's 3 or 4 years, or a master's degree. So this short course aims to fill the gap.
A key motivation for our short courses was to really support our next generation of breeders, and they are our PhD students. We've found that a lot of the undergraduate courses that are available at universities tend to be quite broad, and there's nothing quite specific on plant or animal breeding. So we wanted to provide our students with the fundamentals, the foundations, so they can hit the ground running in their PhD.
Okay. And well, one of the really cool things about that course is that the participants will have to develop their own breeding program, design their own breeding program, which they're going to take into the next course and optimize. So yeah, please, you can actually fill in the EOI for that course, and stay tuned for the launch and more information.
Thanks, Lee. Just a reminder too, you can watch all the seminar recordings online, and you can sign up here with the QR code so that you can get notified by all the different seminars that we got coming up next year in the ARC Training Centre for Predictive Breeding. And I just want to say a big thank you for coming, and I really hope you enjoyed this talk. And again, a round of applause for Lee for this amazing session.